A very good morning to my dear students of class 9. Welcome to biology class. We are dealing with the chapter digestive systems, so we'll continue with this chapter only. So today we are going to learn about the oesophagus and the stomach and the other part of digestive system. So oesophagus, that means this particular portion, okay, the long tube-like structure. So we are going to learn about this particular part. It's called the oesophagus. So oesophagus is mainly a tube which will conduct the food, which will transport the food from the throat to the stomach. Clear? And it passes through the diaphragm, which is close to the backbone. So here you can see these black structures, which is called the diaphragm. Okay, that will mainly separate your gut portions and the lungs. Now, this oesophagus does not produce any type of enzymes because enzymes are essential for the digestion of food particles, but oesophagus does not produce any type of digestive enzyme that will help in digestion. Clear? So once the food will reach the oesophagus, it moves toward the stomach by the action of peristalsis. We have learned in last class also about peristalsis, where there will be the rhythmic contraction and relaxation of the muscles of the gut, so that the food will move towards the stomach from the oesophagus. So by the help of the peristalsis, by the way of constrictions or the contraction which caused by the circular muscles of the gut, the food from the oesophagus will reach towards the stomach. Okay, and peristalsis will occur throughout the region of the gut. So there will be contraction, there will be relaxation throughout the oesophagus. Okay, and finally from the oesophagus the food will reach to the stomach. Clear? Now, what is stomach? The stomach is an elastic bag. Okay, so here you can see the present stomach. Okay, this is stomach part. So here you can see this is the stomach part. Okay, so stomach is an elastic bag located below the diaphragm. So the black one in the figure is the diaphragm, and just below the diaphragm we have got the bag-like structure which is called the stomach. Okay, and mainly in adult it said that it can hold up to 2 to 3 liters of food. Now within the stomach we have got highly muscular layers which has got the property to thoroughly mix the food particles. Okay, and the inner lining of the stomach will also secrete the gastric juices. Remember, the inner lining of the stomach will secrete the gastric juices. Okay, now below the stomach you can see the presence of intestine. Right, so the opening of the stomach into the intestine is called pylorus. Please keep in your mind that the opening of the stomach into the intestine is called the pylorus, and this pylorus has a ring-like muscle called the spinster. Okay, it has a ring-like muscle called the spinster which will keep the opening closed, okay, like a valve, to prevent the food passing from the stomach until it is thoroughly churned up. So this pylorus, which has got the ring-like muscle called spinster, it acts like a valve, okay, in the artery, okay, in the heart, that will prevent the backward flow of the blood. Okay, so similarly in stomach also we have got these spinster muscles which will close the valve so that it can prevent the passing of the stomach until it is thoroughly churned and thoroughly digested. Okay, now this particular spinster muscles, okay, we have got another type also which is called the cardiac spinster. So we have got another type of spinster which is called the cardiac spinster. The function of this cardiac spinster is to prevent the backward flow of the food into the oesophagus from the stomach. Okay, it is present at the front end of the stomach, whereas that pylorus is at the opening where the stomach will open into the intestine. We have got the spinster muscle over there also. Similarly, at the front end of the stomach also we have got the spinster called the cardiac spinster, and you know the function of spinster is to prevent the backward flow of the food. So the cardiac spinster function is to prevent the backward flow of the food into the oesophagus from the stomach. Clear this one? Now, gastric juices. We have learned that the gastric juices is secreted by the inner lining of the stomach, and this gastric juices is colorless, highly acidic liquid containing water, some salt, hydrochloric acid, and an important enzyme called pepsin. Remember that gastric juice contains an important enzyme called pepsin, and this gastric juice has got highly acidic liquid. Okay, so the acid serves two functions. We also got HCl in our stomach, in the gut. Okay, so the acid has two functions. Number one, it will kill the germs which have entered the body entered the gut through the food, and number two, it activates the enzyme pepsin to act on the protein. So without this acid, pepsin cannot be activated. So in the presence of the acid, the pepsin will be activated and will it act on the protein. That means the pepsin will digest the protein into peptides and amino acids. Okay, and mainly when we go into the detailed study of the activation of pepsin, the pepsin is first secreted as pepsinogen. Clear? The pepsin is first secreted as pepsinogen, and that pepsinogen will be changed into pepsin by the action of HCl. So you can go to the book also, page number 109, that pepsinogen will be in the inactive form. Then this pepsinogen will be converted into active pepsin by the help of the acid called HCl. Then pepsin will act on the protein and will change into peptides. Clear? Okay. The food will stay in the stomach for nearly 3 hours, and it entails a pulp-like form called the chyme. Okay, and whenever the chyme is formed, then the spinster muscle will open up, and the food from the stomach will move to the intestine. Clear? Then comes the small intestine part. So okay, now we will talk about the small intestine part. Now, small intestine is a tube of about 7 meters long and 2.5 centimeter wide. Okay, it is coiled and folded in the abdomen, and therefore the small intestine is also divided into 3 parts: duodenum, jejunum, and ileum. So we look over here. So remember that small intestine is divided into 3 parts. First part is called the duodenum. Then we have got the second part of the small intestine called the jejunum, and the third part is called the ileum. So these are the 3 divisions of the small intestine. Duodenum is the sub upper part next to the stomach. You can see the duodenum because it is close to the stomach part. Okay, then we'll say that the common bile duct will open into this part. Then we have got the jejunum, which is the next short region of about 2 meters, and it said that jejunum means empty because it is nearly always empty after death, as found in dissections or in post-mortem. Then we have got the ileum part, which measures about 4 meters, and ileum means to twist or roll, referring to the twisting movement of this part. So we have got the 3 parts of the small intestine: duodenum, close to stomach, then we have got the jejunum, and we have got the ileum. Okay, and mainly in duodenum the bile ducts will open up. Clear? Okay, now ileum part we'll discuss. Okay, because the inner lining of the ileum has a tiny finger-like projections which are called the villi, V-I-L-L-I, villi, or we also call it villus. Okay, now what is the function of this villi, or how does this small intestine will help in the digestion or the absorption of the food particles? Because the inner lining of the small intestine, that is let's say the ileum, has the finger-like projection which is called the villi, and the function of the villi is that it will increase the inner surface area of the intestine for the absorption of food particles. Remember carefully that the inner lining of the ileum contains a finger-like projection which is called the villi or the villus, V-I-L-L-I, villi, also called the villus, and this villi will increase the inner surface area of the small intestine, which will facilitate the digestion, or let's say the absorption of the digested food particles. Okay, and between the villi there is small holes which are called which will secrete the intestinal juice. Clear? And this intestinal juice is poured into the lumen of the intestine. And villus is mainly covered by a single cell layer called the epithelium, and inside the villus we will find the artery, we will find the vein, and we will find the blood capillary and the lymph vessels called the lacteals. We said that the villus is covered by a single layer of cells which is called the epithelium. Okay, and inside the villus we will find vein, we will find artery, we will find capillaries, and we will find a lymph vessel which is called the lactea. So they are all situated in the small intestine. Now the small intestine has got the function of both digestion and absorption. Okay, because it receives 2 digestive juices, and what are those 2 juices which are present in the small intestine? One is the bile and the pancreatic juice. Clear? So both the juices are mainly been received by the small intestine. Now let's talk about the bile. Now, bile is a yellowish green and watery fluid produced in the liver. Remember that bile is produced in the liver, and after its production, it is transported through the hepatic duct. Okay, and hepatic duct is connected to the cystic duct by the help of the common bile duct. So we are saying that bile is produced in the liver. After its production in the liver, it is transported by the help of the hepatic duct. Clear? And hepatic duct is connected to the cystic duct to form the common bile duct. Clear? So you can see in the figure also that we have got the hepatic duct. Okay, we have got the cystic duct. So here you can see the liver part. Okay, it's mentioned here, it's mentioned from liver. So from liver, bile will be produced. That bile will be collected by the hepatic duct, and it will join the cystic duct, and cystic duct and the bile duct will form a common bile duct. Okay, so hepatic duct and cystic duct will together form a common bile duct. Alright, now the bile may flow directly into the duodenum, or it will be temporarily stored in the gallbladder. Yeah, so biles can directly enter into duodenum, or mainly for a short period of time the bile will be stored in the gallbladder. And the color of the bile is due to the presence of a pigment called biliverdin and bilirubin. Remember this one. Okay, the color of the bile depends on the presence of the certain pigment, and those pigments are mainly biliverdin and bilirubin. These are the name of the pigment which are present in the bile, which are produced by the breakdown of the dead and the worn out RBC cells. Bile also contains a lot of sodium bicarbonate, and this will neutralize the acid content of the food from the stomach and make it alkaline, so that the pancreatic and the intestinal enzymes will act on the food. Then next one is bile salts will reduce the surface tension of the fat and will break down into tiny droplets for providing greater surface area for the enzymes. Overall, the function of the biles, we can say that, is to convert the acidic chyme into alkaline chyme, because acidic chyme becomes alkaline by the presence of sodium bicarbonate. And next function of bile is to go for emulsification, where the fat will be changed into the emulsified fat. Okay, these are the general functions of bile. The next one we talk about pancreatic juice, which is produced in a white gland called pancreas, located behind the stomach. Okay, so behind the stomach you can see the white gland, which is called the pancreas. So this pancreas will secrete the pancreatic juice, and pancreatic juice will open into the duodenum by a common aperture to that of the bile duct. Okay, so this pancreatic juice has got three important types of enzymes. Very important. Pancreatic juice contains three types of enzymes, that is amylopsin, trypsin and steapsin. Clear? So it has got three kind of enzyme: amylopsin, trypsin and steapsin. Sorry for the figure, you can go to the page number 110 also is there. Okay, so amylopsins, it will digest the leftover starch into maltose. Generally, mainly in the mouth, the saliva contains the enzyme called peptidine or the salivary amylase that will convert the starch into maltose. Clear? But the leftover starch will change into maltose by the help of amylopsin. Okay, then trypsin will act on the remaining proteins and polypeptide to produce smaller peptide and amino acids. And like pepsin also, trypsin is first synthesized as inactive trypsinogen and is converted to active trypsin by the enzyme called enterokinase. Clear? Okay, then third one is steapsin, which will act on the emulsified fat to split them into fatty acids and glycerol. So my dear, remember the end products of protein, fat. Protein will break broken down into peptides and amino acids, fat into fatty acids and glycerol by the help of the enzyme steapsin. Clear? Okay, so trypsinogens will be in inactive form. It will be converted into active form trypsin by the help of the enzyme enterokinase. Then that trypsin will act on the proteins and the peptides to form smaller peptides and amino acids. Leftover starch will be acted upon by the enzyme called amylase or amylopsins, or also called pancreatic amylase, and will change into maltose. And emulsified fat will be acted upon by steapsin and will change into fatty acids and glycerol. So kindly remember the end products of protein, fats and the leftover starch also. Clear? And the name of the enzymes which are present in the pancreatic juice. The intestinal juice contains an enzyme called erepsins. okay, contains erepsins, which is mixture of peptidases, and it will convert the remaining peptides into amino acid, maltose to digest maltose into glucose, lactase to digest lactose. Listen very carefully the name of the enzyme. Erepsin will convert the remaining peptides into amino acid. Maltase, M-A-L-T-A-S-E, maltase will digest the maltose into glucose. Then we have lactase, L-A-C-T-A-S-E, lactase will digest the lactose into glucose and galactose. Sucrase, S-U-C-R-A-S-E, sucrase will digest your sucrose into glucose and fructose. And there are traces of lipase, L-I-P-A-S-E, lipase, which will digest the fat into fatty acids and glycerol. So all these enzymes are present in intestinal juices. So pancreatic juice contains enzyme amylopsin, trypsin and strepsin, and your intestinal juice contains the enzyme erepsin, maltase, sucrase, lipase and lactase. Clear? So remember the name of the enzymes and in which particular juice they are present. Now we'll go into the next one, which is called the absorption of food. So after the digestion of food, after the final products of food are mainly absorbed in the intestine itself. So when the food products will be digested, now they have to be absorbed, and the absorption of the food mainly occurs in the intestine. So the amino acids and the simple sugar, okay, which are relatively small size molecules, they are absorbed through the epithelium of the villi. Listen very carefully. He said that the small size molecules are absorbed through the thin epithelium of the villi, and they reach the blood capillary to finally enter the blood circulations through the hepatic portal vein. Hepatic portal veins means the vein that goes to the liver. Clear? Fatty acids and glycerols are absorbed to the limb vessels or the lacteals. Remember this thing. Fatty acids and glycerols are absorbed by the lacteals and they will enter the lymphatic systems. Clear? Okay, then the food passes very slowly from the intestine, which takes about 4 hours to enter the last part, which is called the large intestine. And my dear, large intestine is about 1.5 meter long and it has 3 parts: caesium, colon and rectum. So we'll see the part of the large intestine. We have got the large intestine part, which has got caesium, okay, we have got colon and the rectum. So this part is the large intestine part, divided into caesium, colon and rectum. Okay, now let's see the function of caesium. The caesium is a small blind pouch situated at the junction of small and the large intestine. So the junction of the small intestine and the large intestine, we will find what? We will find caesium. So this one is the small intestine, appendix part, the small intestine. So the junction, so at the junction of the small, at the junction, okay, so the junction of the small intestine and the large intestine, we have got caesium. Clear? Okay. Now from its blind end projects a narrow worm-shaped tube called the vermiform appendix. Remember, this vermiform appendix is a vestigial organ. It was functional in our ancestor, but at present time it is non-functional. Okay, its function was to digest the cellulose which is present in raw plants that our ancestors used to eat. But nowadays we used to eat the well-cooked food particles. That's why there is no need of appendix, and it remains in the form of vestigial organ. And whenever this appendix will undergo inflammation, it will cause a disease called appendicitis. But today it has remained in the form of a functionless organ. It's called the vestigial organ. Then comes colon, which is broader than the ilium and is a little more than a meter long. It passes from the abdomen on the right and crosses to the left just below the stomach, okay, and down the left side, descending colon. That means if you look at the colon 1, so we have got this one from the right side, okay, it's called ascending colon. Then it just crosses below the stomach. You can see this is the stomach part. It just crosses below the stomach. Then we have got the ascending, and then we have got the ascending colon. Okay, then we have got the colon that passes just below the stomach. Then we have got the descending colon on the left sides. So this one is the ascending colon, and this just passes through the stomach, this below the stomach. Then we have got the descending colon. And last part is the rectum. The rectum is the last part, which is about 15 centimeter long, and it opens at the anus. And the anus has a muscular muscle called the spinster, which helps it to close except when passing bowels. So whenever we are passing the bowels, when we are passing the waste materials, the spinster with the anus will open up. Otherwise, it will usually close. Clear? Then from your book, my dear, kindly go through the table number from your book. Okay, from your book, kindly go through the table number 11.1, where you will find the summary of digestion in various parts of the human alimentary canal. Okay, I am saying that it's in the page number 111, table number 11.1. The topic is summary of digestion in various parts of the human alimentary canal. So kindly go through it. Alright, now we will talk about the functions of large intestine. The large intestine also does not secrete any type of enzyme, but will absorb much water but very little digested food materials. Okay, after much water is absorbed, the contents before semi-solid phases, which passes to the rectum, becomes, listen carefully, that large intestine is absorbing water. It is absorbing much more amount of water than digested food. So after water will be absorbed, the contents, the food materials, okay, after absorptions become solid phases, and it passes to the rectums, and from the rectum it passes to the anus, and from the anus the sphincter muscles will open up, and that fecal materials will be excreted. Okay, and that is called defecations. So the expulsion, the removal of the undigested remains of the food from the alimentary canal is called the defecation. Okay, and the feces are normally composed of water 75% and 25% solid matter, which again consists of 30% dead bacteria, 10 to 20% fat, 2 to 3% protein, and 30% roughage. And the bad odor of the feces is due to the bacterial action in it. Okay, so this is mainly the composition of our fecal materials, the material that we used to excrete. Clear? Next one, we'll talk about the assimilation of food. So assimilation means the conversion of the absorbed digested food into the body materials. Okay, so we have got digestion, we have got absorption, and after the absorption of food materials, it has to be utilized in the body cells, which is called assimilations. So the food digested and absorbed by the gut are transported in two ways. One is through the hepatic portal system, and the one is through the lymphatic systems. Okay, so simple sugar, amino acid, vitamins, minerals are carried to the liver by the hepatic portal vein. Hepatic means liver. Okay, and the liver convert the excess glucose to glycogen. Very carefully, the liver will convert the excess glucose into glycogen by the process called glycogenesis. Clear? What is glycogenesis? The process of conversion of excess glucose to glycogen is called glycogenesis. And whenever there is a demand for the glucose in the body, now that glycogen will be again reconverted back into glucose, which is called glycogenolysis. Clear? So excess glucose will be changed into glycogen in the liver by the action class glycogenesis. But whenever there is a demand for the glucose, now the glycogen will be converted back into glucose, and this process is called glycogenolysis. And now glucose will be put back into body circulation. Okay, and glucose is a source of energy in our body. Now next one, the amino acids circulate in the body, and they serve as the building block of the protein, because amino acids, they are the building block of the proteins. Okay, so amino acid cannot be stored. Any excess amino acids will be broken down in the liver by the process called deamination. Clear? In which the nitrogen groups containing amino acids will be removed, and it will be converted into urea for excretions, and the remaining part forms the glucose, which can be utilized. So amino acids says that amino acids are broken down in the liver by the process called deaminations. So those amino acids which contain the nitrogens, they will be changed into urea, and the remaining part will be form the proteins. They will form the glucose, and that glucose can be again reutilized. Okay, now fatty acids and glycerols, which are the end products of fat, they are absorbed by the gut, and they are transferred to the intestinal villi and the lymphatic systems. Clear? Okay, now fats, they are used in the synthesis of certain compound in the body cells, and the excess quantity is deposited chiefly below the skin as subcutaneous fat or around the certain visceral organs in a smaller quantity. Now let's go to liver. Now liver is the largest gland of the body. It's weighing about 1.5 kg. Okay, this reddish brown organ located in the upper right side of the abdomen, just below the diaphragm. Now from your book, I would like to tell you to just go through the function of liver. Okay, because liver, they used to control the blood sugar level by changing or by converting the excess glucose into glycogen. They control the level of amino acids by the process called deaminations. They will help in the synthesis of fetal red blood cells. They will help in the storage of iron, copper, okay, and several vitamins like A and D. They help in detoxification. They detoxifies the substances including drugs and alcohols. They will help in the production of fibrinogen and prothrombin, which are essential for blood clotting. They help in the production of heparin, the natural anticoagulant present in the blood. They help in the production of heat, and it helps in regulation of blood volume. It will help in the excretion of toxic and metallic poisons, and will help in destruction of red blood cells also. So kindly from your book, page number 113, go through the function of liver. Okay, now we will learn the certain experiment on digestions, but the most important one we are going to learn is the action of pepsin, the enzyme that may digest the protein. Okay, so we will go to point number 2, that the action of pepsin on the egg white protein. So what we do to process an experiment, we will stir the white of an egg in about 500 ml of water. So around 500 ml of water, we will take the white portion of the egg and we will stir it. Okay, then boil the mixture. That means we have to boil the mixture. Mixture means that 500 ml water containing white part of the egg. So we have to boil it. So when we used to boil it, and when we filter it through the glass hole, are you getting it? We have to boil the mixture. Mixture means 500 ml water along with that white portion of the egg. Okay, so we are boiling it, and after boiling we are filtering it. Now the filtered will be a cloudy liquid suspension. Clear? Okay, so we have filtered the boil part and we are getting the filtered, which have a cloudy liquid suspension. Now what we do, we will take 4 test tubes and we will mark them A, B, C and D. Okay, we are taking 4 test tubes and we are marking them A, B, C and D. So this is what we are doing. We are taking the white portion of an egg in 500 ml of water. We are boiling them. After boiling it, we are going for filtrations, and now the filtrate will be collected and it will have a cloudy liquid suspension. After that, we are taking 4 test tubes. We are marking them A, B, C and D. Clear? Okay, now take a small quantity, about 3 ml of suspension in each test tube. Now that filtrate which we have done from the mixture, okay, that filtrate has to be added in all the 4 test tubes. Nearly how many ml? 3 ml. So 3 ml of filtrate, that cloudy liquid suspension, has to be added in all the test tubes. That means 4 test tubes. Now after that, we'll add a few drop of pepsin and hydrochloric acid as follows. Okay, so in test tube A, we'll only add pepsin. In test tube B, we will put only HCl. In test tube C, we will put pepsin plus HCl, and in test tube D, we will put HCl plus boiled pepsin solution. So we are taking 4 test tube. In the 4 test tube, we are putting the suspension, okay, that filtrate part, and we are putting the enzyme and the acid. So in test tube A, we are putting only pepsin. In test tube B, we are putting only HCl. In test tube C, we are putting pepsin plus HCl, and in test tube C, in D, we are putting HCl plus boiled pepsin solution. Clear? And we'll place all the test tube in a beaker of water, and we will keep them at 35 degrees Celsius for nearly 10 minutes. So we'll boil the water of 35 degrees Celsius, and for nearly 10 minutes we'll keep all the test tube over there, that we are trying to maintain the temperature for the enzyme to act. Okay, now which of the test tube among the 4 will form a clear solution? Because the filtrate was cloudy suspension, okay, with contained proteins. But now out of the 4 test tubes, which one will show the clear solution? Clear solution means that protein will be digested. Okay, so which of the test tubes will show the formation of a clear suspension from a cloudy one? So when we look at the 4 test tubes, you can see that test tube A contains only pepsin. So test tube A does not show clear solution. Test tube B contains only HCl, there is no enzyme, so it doesn't show the clear solution. But test tube C, it contains pepsin plus HCl, and if you try to remember that pepsin will be activated in presence of acid. So here in test tube C, we have got pepsin we have got HCl. So HCl will activate the pepsin, and pepsin will digest the protein. So in test tube C, the cloudy suspension will change into clear solution. Clear? So we can say that pepsin can digest protein only when it is acidic. Acid alone has no effect on protein. But in test tube D, we are putting HCl, we are putting the pepsin, but that pepsin was boiled in test tube D. So therefore boiled pepsin will fail to digest the protein, because when we are heating the protein, it will become denatured, it will become non-functional. So the boiled pepsin solution will not be able to digest the protein. In other words, the enzyme pepsin is destroyed by the heat. So therefore only the test tube C will show the formation of clear solution from the cloudy solution. Clear this one? Okay. Then next one is the number 3, shows the effect of acidity and alkalinity on an enzyme reactions. So in the same experiment with the white part of the egg that contains protein, we are going to study the effect of acidity and alkalinity. Okay, so what we do, we'll take the 5th test tube and we'll mark it E. Clear? With the same experiment, we take the 5th test tube, that is E, in which we take the egg part suspension, and in that part we'll put the small quantity of pepsin, and we'll put the small solution called small drops of mild sodium bicarbonate solution. Listen me carefully. We are taking the test tube E in the same experiment. In the test tube E, we are putting the small, let's say 3 ml of suspension we are putting in test tube E. Okay. Then don't get confused. In the same experiment, we are taking A, B, C, D. Right, we are doing the experiment to check the action of pepsin on the protein. Now in the same experiment, we are taking the 5th test tube, we are marking it test tube E. Again in the test tube E, we are putting that solution, cloudy solution. Okay, then we are putting pepsin. Then we are putting the mild sodium bicarbonate solution. In place of HCl, we are putting mild sodium bicarbonate solution to make it alkaline, because we know that pepsin will be activated in presence of acid HCl. But now what we are doing in test tube E, we are putting the solution, that is cloudy white. Okay, we are putting the pepsin and we are putting the mild sodium bicarbonate solution. This mild sodium bicarbonate solution will make it alkaline, and again we'll keep it in that water for 10 minutes at 35 degrees Celsius. There will be no change in the the cloudiness of the solutions. Why? Because pepsin does not act in the alkaline medium. Pepsin does not become activated in case of alkaline medium. Pepsin will only become activated when there is hydrochloric acids. Clear? So pepsin becomes activated in presence of acidic medium, but pepsin doesn't become activated in presence of alkaline medium. So here we have finished a few experiments on digestions. Now we are going to just check out the practical work on food test. Okay, just listen carefully. We'll first check the test for glucose. And over here, one thing I remember is that you have to remember the chemical that we are going to use for the experiment and the change in the color. Okay, so first we will test for the glucose, for the sugar. So a little glucose will be added to a blue-green chemical called Fehling solution. So my dear students, kindly remember the name of the solution, Fehling solution, which is used for the test of glucose. Okay, so a little glucose will be added to a yellow-green chemical called the Fehling solutions in a test tube. Clear? Then gently tilt the test tube away from you and heat it in a flame. Okay, until bubbles begin to appear, but not allowing it to boil over. Now when this will happen, what we'll see is that the color will change from the blue-green to a deep brick red with appearance of precipitate. This is very important. The color will change from the blue-green to a deep brick red with the appearance of a precipitate. So what we are doing, we are putting a little glucose in a yellow-green chemical called, in a blue-green chemical called Fehling solution test tube. We are tilting the test tubes and we are heating it. Okay, but we are not allowing it to overboil. So when we are heating it over a flame, what we observe, we will indicate a change in color. The blue-green color will change into a deep brick red color with the appearance of precipitate. Clear? A small piece of apple or grape will give the same result. So when we are going for the test of any fruit that contains sugar, the glucose, we will find the same changes. We have to use the Fehling solution, that is blue-green in color. We have to put the glucose over there. We have to tilt it. Okay, we have to heat it on flame, but we don't have to overboil it. So when we do it, we will observe the change in color from the blue-green to deep brick red color with the precipitate, and this will indicate the presence of sugar. Okay, the next one, test for starch. How can we prove that the given sample contains starch? So little starch powder is taken in a test tube with some water, and we have to boil it. We have to make a solution. Okay, and when the solution will be cold, we have just add a few drops of iodine solution. So iodine solution is used to test the presence of starch in a given sample. Clear? Now the solution will be diluted before using. This is that the starch solution will turn blue-black, indicating the presence of starch. So the color will be what? It will be blue-black. The starch solution will turn blue-black, indicating the presence of starch. So what we are doing? We are taking the starch powder, okay, in a test tube. We are adding some water. We are cooling it. Clear? And we are making solutions. So after cooling it, what we are adding? We are adding the iodine solutions. When we add the iodine solution, and if the specimen contains the starch, then the starch solution will turn blue-black, indicating the presence of starch. Next one, test for protein. How to prove it? So place a piece of hard-boiled egg white in a test tube. So that place a piece of hard-boiled egg white in a test tube. Then we have to add a few drops of concentrated nitric acid. Clear? Enough to immerse the sample of the food taken. Then heat the test tube gently. Then cool the test tube by pressing it under tap water. And what we are adding ammonium hydroxide. So when we add the ammonium hydroxide, we will notice a color change. First, from the white precipitate, it will change to yellow upon heating, and then from yellow to orange upon adding ammonium hydroxide. So remember the color change. What we are doing, we are putting a piece of hard-boiled egg white in a test tube. Okay, then we are putting a few drops of concentrated nitric acids so that it can completely immerse the food particles, and we are heating the test tube. And then cooling, we are putting ammonium hydroxide. So when we are doing this one, we will indicate or will observe the color change. First, it says that the color will change from white precipitate to yellow, and on cooling, the color will change from yellow to orange when we add ammonium hydroxide. So this will indicate the presence of protein in the given sample. Okay. Then you have a test for fats and oil. Just drop a piece of groundnut, walnut, or a piece of butter on a plain paper. So just take a groundnut, walnut, or a piece of butter, and we drop it on a piece of paper, plain paper. The place where we have rubbed that groundnut, walnut, or butter will turn translucent, especially when viewed against the light. Okay, so here we have finished the chapter digestive systems, and from the next class we'll begin with the new chapter. So this much for today. Thank you, and see you in next class.